I'm the CSO of Abterra Biosciences, and I'm going to talk to you today about next generation repertoire sequencing for mining single domain antibodies from Camelid repertoires. So, before I delve into that topic, I just wanted to give an introduction about Abterra and who we are. So, we were formerly known as Digital Proteomics, and we recently changed our name to Abterra Bio to reflect the fact that we focus on antibody sequencing, characterization, and discovery using high throughput sequencing technologies such as next generation sequencing and mass spectrometry proteomics. So today I'm going to talk to you about our proteogenomic discovery approach, which utilizes both of those underlying technologies, NGS and mass spec. And as part of that, it's going to touch a little bit upon uh, repertoire, which is what we offer for next generation repertoire sequencing of cellular repertoires, as well as some parts of valence, which is our resequencing of monoclonal antibodies when there is no cellular material available, no genetic material. So it's only from proteins. The case study we're going to be talking about is in camelids, so in particular llamas, but this is applicable to camels as well as alpacas. So camelids produce unique antibodies in that they produce a conventional antibody, which has a heavy chain and a light chain. And in llamas, this is produced as an IgG1 isotype. But they also have this unique heavy chain only antibody, which is devoid of the light chain and only has a heavy chain. And in llamas, this is produced as an IgG2 or IgG3 isotype. And one can express just the variable region of this heavy chain only antibody as a VHH. And VHHs have nice properties associated with them. Namely, they're easy to express, such as in bacterial systems. They're stable under several different conditions. They're small. They're modular, so they're more amenable to protein engineering, and they can target concave epitopes that conventional antibodies can sometimes struggle to do. So current approaches to discovering these single chain antibodies typically rely on phage display, but phage display has certain drawbacks associated with it, namely that you can result in libraries with lower diversity than what is seen in naturally occurring repertoires. And this is related to the difficult biopanning steps that are needed for panning. So the case study today is gonna be highlighting some work we did with a llama immunization against a soluble protein antigen, which was boosted several times over the course of a 14-week immunization schedule. As part of that, we collected PBMC samples from blood at 8 different time points, and finally, we collected a serum sample at the end of the immunization schedule. And this serum sample is really important as it drives our discovery approach. So this serum sample was affinity purified against our target of interest to select out the heavy chain only antibodies that are reactive to that target. This was then subjected to mass spectrometry proteomics, and this is what we use to do the discovery. So that was basically this top tract of our Alicanto approach, where we have the serum proteomics track up top that purifies and focuses what was in the serum that's reactive to our target of interest, that is a heavy chain only antibody. This bottom track is the next generation sequencing of the llama repertoire that we're going to utilize as part of that as well. So I'm going to talk about that now. So llama repertoires are a little different than other types of repertoires that may be more familiar with, such as human and mouse. Human and mouse repertoires typically have fairly diverse V gene usage. But as you can see here, this is the V gene usage in the llama across all the different time points that we sampled. And it's very V gene focused. So in the top, we see the G2 isotype, and about 75% of the V gene utilization is from a single scaffold. So it's very focused. And on the bottom is a G3 isotype. And you have within the top three V genes, they account for about 75% as well. So it's also quite focused. So the llamas and alpacas as well have a small set of favored scaffolds that they use. There's some other differences as well. The G2 versus G3 mutation rates differ a little bit. So G3, seen here in orange, is more divergent from the germline than G2 is, which is seen in blue. And this plot up here is an amino acid mutation distribution, and the bottom is the same repertoire, but the mutation distribution is seen in nucleotide space, so it's even more pronounced. And this is shown for week 10 PBMCs, the difference. And you can look at the differences between any of the different time points, and they're very similar. There is a divergence between G2 and G3, and G3 tends to be more divergent from germline. This also persists across different immunizations, though we're not going to show that. Finally, we also look at positional mutations in the amino acid space. So this is for week 10 G2 library for the favored scaffold, and we can plot in the stacked bar plot all of the amino acid mutations that were observed in the repertoire, the next generation sequencing repertoire. And what we see here is, unsurprisingly, that CDR1 and CDR2 are more highly mutated than the rest of the antibody. However, the framework regions are still fairly mutated, and there's some mutations that are more common than others as well. So if we focus in on CDRs 1 and 2, as is commonly done, we can look at the the frequency distribution of CDRs 1 and 2. So this is CDR1, and at the top is this entropy plot, where entropy is a proxy for diversity at that particular position. And so what we can see is that there's two positions in CDR1 that are more divergent, more diverse, than the remaining positions. So not all positions are created equal, even within CDRs. And this trend also continues in CDR2, where there's about 4 or 5 different positions that are more diverse than the remainder of the CDR. And this can be important in selecting candidates, in comparing pre-panning and post-panning libraries, or pre-immune and post-immune libraries, or if you're creating an in silico library, maybe you want to capture these specific positional diversity hotspots. So these summary distributions about the next-gen sequencing repertoire are helpful for comparing the repertoires or subsets of repertoires, but in the end, we want individual clones and antibodies for discovery purposes, for actually testing out. So that's when we incorporate the proteomics that I mentioned earlier. So we take this next-gen sequencing repertoire, and now we're going to add the target-specific proteomics. And so you can think of the use in the proteomics in this way as to filter the very large next-gen sequencing repertoire that we have through the lens of the proteomics. So if this is a cartoon diagram of the large cellular repertoire and the more restricted serum repertoire, this intersection is where proteogenomics lies. It's going to be the intersection of the next-gen sequencing and the proteomics. And so one way to look at these kinds of data is to identify which clones in the next-gen sequencing are present in the proteomics. So one way to do that is with this spawning plot, where every row is a different serum-identified clone, and every column is a different next-gen sequencing time point. And we can track, using this, we can track clones across the time point sampling, and we can also look at which time points produced an explosion of new clones, since these are ordered by the first occurrence of each clone. So the other thing we need to do is, once we have all of these serum identified clones, we need to select a subset of them for moving forward in the discovery phase. So what we want to do is look at them in this edit distance matrix of the CDR3 sequences. We do this so we don't overpick and make our candidates too homogeneous. So you can see here is we don't want to pick all our candidates in this one cluster. For example, we want to spread it out across all of the different clusters. And sort of to ensure this, we take this matrix and we map it into 2Ds and color the points according to whether they're a candidate, such as orange, or if they were left out of the candidate set in blue. And this is just a way of making sure that the candidates that we picked cover the CDR3 space in a fairly uniform manner. So it's diversifying the candidate portfolio. And then finally, we also look at the individual proteomic support for candidate sequences. So this is a candidate sequence up at the top, and each of these rectangles are all of the different unique peptides that were identified in the serum proteomics and how they map back to each individual candidate. So you can see the level of proteomic support over each region, like the CDR3 region, or a specific variants as well, which can be important to provide confidence in calling. So that's how we select candidates from serum. But can we use this next generation sequencing repertoire to extend those hits? So, like I mentioned, Alicanto uses the proteogenomics to identify hits from serum repertoires, this restricted serum repertoires. And other approaches also look at restricted repertoires. So hybridoma hits are from restricted fusion repertoires, single cell hits are from restricted subsampled repertoires, and display approaches also are from restricted pan repertoires. So it's part of the screening approach itself to focus in on a focused, restricted repertoire. But what's that leaves out a lot of information that's still retained in the next generation sequencing repertoire, which captures more members of the same families, more members of the same clonal families of those hits. So can we leverage this? So bringing back this cartoon diagram of the large cellular repertoire and the serum repertoire, the proteogenomics that we talked about it's in this intersection, and we want to push out our hits into the parts of the larger NGS repertoire that were not as well covered by the proteomics. And this is no different than instead of using the proteomics to use hits that were obtained from single cell or hybridoma to expand those out. So how do we do this? We look at the clonal family trees that are in the next-gen sequencing repertoire for our hits. So this is an example tree from the same immunization campaign as and contained one of our candidate hits. So this is a tree that's rooted at the germline V gene. So this is the common scaffold. And each square is a observed sequence in the NGS repertoire. And the size of the square is also proportional to the abundance in the NGS repertoire, but that's maybe less important right now. So if we have a hit, can we find better siblings of that hit? And to test this out, we selected several different candidates from this clonal family to express and test and see, based on the topology and sequence, they were selected to try and cover the space, the topological space, pretty evenly. And so of all the ones that expressed and showed signal, we found our hit, which was already pretty good, and we found several siblings that were better than our proteogenomically identified hit, as well as several that were worse. And going back into the topological structure and the sequencing, we can throw out considerable number of these potential candidates, siblings, based on conservation as well as topology, and then rule in some additional a smaller set of likely better performing candidates based on those same criteria. And the point of this is that this was not a one-off of this was just a good example. This examples of this occur in the NGS repertoire. There's a forest of other possibilities for every uh, clonal family that was sampled deeply, and many of these do overlap existing hits. So in summary, we went over our proteogenomic repertoire discovery that utilizes serum for focusing the next-gen sequencing repertoire on uh, target-specific candidates. I also talked a little bit about how we leverage the large NGS repertoire to improve these within family hits. So this is finding better affinity candidates within the families, and this is regardless of the hit source. So we showed it for our proteogenomics approach, but like I mentioned, you can get a hit seed from any approach, be it hybridoma, single cell phage, etc. And as long as there's a companion next-gen sequencing repertoire, you can do this hit expansion. And what I wasn't able to talk about, but would love to talk about if anyone reaches out, is how we've been working to mine both the next-gen sequencing and serum repertoires independently to extend across family hits. So this was not shown, but again, reach out to us if you're interested. But the idea is that by looking at either similar CDR3 sequences, which are by sequence similarity, or by physicochemical property similarity, meaning they're more divergent than sequence alone, how to extend into those clonal families to find additional candidate hits, companion hits. So that's pushing out more into this NGS repertoire across families. And we can also push across families in the serum only repertoire by doing de novo based discovery and folding those back into the next gen sequencing. So with that, I want to thank all of you for listening, and I wanted to thank everyone at Abterra Bio, but particularly Tiago Lima, who did all of the wet lab experimentation that was presented today, as well as everyone else at Abterra that helped with the remainder of the work that was presented. I also want to thank the organizers at PEGS, and also I want to thank all of our customers for allowing us to be part of the discovery process. So if any of this piqued your interest, please reach out to us at abterrabio.com, or if you know us as Digital Proteomics, you can also reach out to us that way. That's great. Thanks. Bye.